Hello, dear student. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, in today's lecture, we are going to talk about a topic that is replication of DNA, or replication means copying. So, copying of DNA, or how DNA copies make a copy of its own, that we are going to study today in the chapter that is molecular basis of inheritance. So, while proposing the double helical structure of DNA, Watson and Crick had immediately proposed a scheme of replication of DNA. And in scheme of replication of DNA, you can see that double-stranded DNA is there, and this double-stranded DNA gets separated into single strand to carry out a replication. So during this replication scheme or during this replication of DNA, okay, if we talk, how this happens, that we need to understand. So first we are going to understand the process. So first if we talk, then the scheme which was proposed by Watson and Crick, that suggested that the two strands would separate and act as a template for the synthesis of a new complementary strand. And after the completion of replication, each DNA molecule would have a one parental and one newly synthesized strand, and that is known as semi-conservative DNA of replication. But first, we are going to talk about machinery and the enzymes and how this particular replication process work. So if we talk about DNA replication, which is also known as copying of DNA, and in this copying of DNA, DNA synthesis take place. DNA synthesis here take place. And DNA synthesis here, polymerization of nucleotide take place. And when a polymerization of deoxyribonucleotide take place, the DNA is going to be synthesized. And for making a copy or a polymer of this deoxyribonucleotide, the one DNA strand act as a template, and based on the this template strand, the complementary DNA strand is synthesized by polymerization of this deoxyribonucleotide. Now here for this DNA polymerases, which are the enzymes, are required. And first we are going to talk about prokaryote, and then we will talk about eukaryote. Okay, so DNA polymerase is here going to synthesize the DNA by using the DNA. So these are DNA dependent DNA polymerases, which are going to use a one strand of DNA and going to synthesize another strand of DNA by using this one strand. And in prokaryote, if we talk, in prokaryotes or bacteria, three types of DNA polymerases are present. And these three types of DNA polymerases, if we talk, that are DNA polymerase 1, 1, DNA polymerase 2, and DNA polymerase 3. Such type of three polymerases are there. And this all polymerases, this all DNA polymerase have exonuclease and polymerase activity. Exonuclease and polymerase activity. Okay. So here exonuclease means a removal of nucleotide from the ends. So this exonuclease activity means a removal of nucleotide from the 5' end or a 3' end. If it start removing a nucleotide from the 5' end of the DNA strand, then and it move towards the 3' end, then the activity is 5' to 3'. But if it starts removing a nucleotide from the 3' end and move towards the 5' end, then it is a 3' to 5' exonuclease activity. Same way, polymerization or a particular joining of deoxyribonucleotide if occur in a direction from 5' to 3', then it is a 5' to 3' polymerase activity. Okay. Likewise, we have to add a polymerase and exonuclease activity. So if we talk here, then DNA polymerase are of three type and both have an exonuclease activity. Suppose if we take DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. Now this all three have a exonuclease activity. And also they have a polymerase activity. Okay, so exonuclease activity and polymerase activity are that. If we talk about exonuclease activity or DNA polymerase 1, it is also named as Kornberg enzyme. Now this, if we talk about exonuclease activity, it has 5' to 3' and 3' to 5' exonuclease activity. Means it has a capacity to remove nucleotides from the free OH end, that is a 3' end, and it has the ability to remove a nucleotide from 5' as well. But the polymerase activity or joining of nucleotide is in a direction from 5' phosphate free end to the towards a 3' end. And here it adds 1000 nucleotide per minute. Okay, 1000 nucleotide per minute. Whereas if you talk about DNA polymerase 2, they have a 3' to 5' exonuclease activity. And polymerase activity is 5' to 3'. And here the rate is 50 nucleotide are added per minute. So here polymerase activity in the same direction, that is 5' to 3', but here the rate is less, that is of 50 nucleotide per minute. But if we talk about DNA polymerase 3, it has a 3' to 5' exonuclease activity, means it remove a nucleotide from the 3' end. Suppose this is a two double stranded DNA. Okay, and this has a 5' end, and this is a 3' end, this is a 3' end, and this is the 5' end. If the enzyme start removing a nucleotide from the 5' end and towards the 3' end, then it is a 5' to 3' activity. But if the particularly enzyme start removing nucleotide from the 3' end and it move towards the 5' end, then it is a 3' to 5' exonuclease activity. So here DNA polymerase 3 have a 3' to 5' exonuclease activity, and here 5 dash to 3 dash polymerization activity rate that is rate that is 2000 base pair per second. 2000 base pair per second. 2000 nucleotide RNA per second. So if we compare the polymerase activity, DNA polymerase 3 is looking the best rate. It is showing the best rate of addition of nucleotide per second. So DNA polymerase 3, if we talk, it is considered as the main polymerizing enzyme. It is considered as a main polymerizing enzyme. Okay. So this is the three polymerases, how they function. Now after this three polymerases, now we are going to talk about the particularly the how the replication occur. So if we talk about that, the process of replication in living cells, if we require a set of enzymes, and the DNA polymerase is discovered, which are the main one. And the main enzymes, these all are DNA dependent DNA polymerases. It is highly efficient with the ability of polymerize 2000 base pair per second in the DNA polymerase 3. Now, it is not that this DNA polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy. And any mistake during replication would result into mutation. So that mutation need to be recovered. That is also one of the important thing. Now, first thing is that the DNA polymerization starts from the, if suppose this is a strand of DNA, it is a, we can say, or we can say it is a coiled DNA. Suppose it is a coiled DNA. Okay, if this is the point from where the DNA replication is going to start, or in a DNA strand, if it is a linear, it is the point from where the particular DNA replication is going to start, then it is known as ORI C. And this ORI C is nothing but it is the origin of replication. Now here, origin of replication. So replication begins at a particular region of DNA, which is called a origin of replication. It is because of the requirement of origin of replication that a piece of DNA, if needed to be propagated during a recombinant DNA processor, requires a vector. So here, dear students, I want to say, or you need to remember that original replication is the point, or it is a particular region of DNA, particular region of DNA from where from where replication begins. From where replication begins. And if we talk about this in case of prokaryotes, then prokaryotes have single origin of replication. Single origin of replication, and it is called a ORIC in E. coli. Whereas on the other hand, in eukaryotes, if we talk, they have several thousands of several thousands of origin of replication. So it is the point from which the origin a replication is going to start. And in replication, if origin occurs, then there is a need of what? A deoxyribonucleotide to add in a non-template strand complementary to the nucleotides present in a template strand. So there is another thing which is needed, that is activation of deoxyribonucleotide. Activation of deoxyribonucleotides. Now there are four types of deoxyribonucleotides. Dear students, four types of deoxyribonucleotides are there, and they are namely DAMP, that is deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxythymidine monophosphate, and deoxycytidine monophosphate. Like this, four types of particularly deoxyribonucleotides are there which are activated by a phosphate energy and enzyme phosphorylase. Okay, these four nucleotides are there. These four types of deoxyribonucleotides are activated by phosphate energy and enzyme phosphorylase. So here, if we take DAMP, DGMP, DCMP, and DTMP, and we react all with the two molecule of H2PO4, that is phosphoric acid, in the presence of enzyme phosphorylase and energy, it is going to form DATP. Why? Because here it is adenosine monophosphate. But when two phosphate molecules are added, it is going to convert into the DATP. Then it is going to convert into DGTP, DCTP, and DTTP. Like this, convert into the four nucleotides. Okay. And this way, the four, particularly DATP, DGTP, DCTP, and DTTP, are produced. Now here, this activated nucleotides or activated deoxyribonucleotides, if we talk, they have two functions. Now here, this activated ribonucleotide, if we talk, that activated deoxyribonucleotide, if we talk, has two functions. It plays two functions, and one function is it act as substrate. It act as a substrate, and second, it provide energy for polymerization reaction, because two terminal phosphate, the last two phosphate of DATP, DGTP, DCCTP or DDTP. In a deoxyribonucleotide, in a deoxyribonucleotide, triphosphate are high energy phosphate, same as in case of ATP. Okay, so this is the two things or two functions if we say for this activated deoxyribonucleotide are used. Now after this there is a mechanism of this particular DNA replication. And if we talk about a mechanism of DNA replication, it occurs in following way. First, what happens? There is a double-stranded DNA. Suppose like this. It is a double-stranded DNA. And here it is one strand is in 3 dash to 5 dash direction and another strand is in 5 dash to 3 dash direction. This strand which is in 3 dash to 5 dash direction, it acts as a template, so we are calling it as a template strand. And this strand it is known as non-template strand actually in this DNA. But if we talk about DNA replication, here both strand are going to act as a template strand. So when both are going to act as a template strand, here first what happens, the double helical, this parental DNA molecule, where helicase enzyme, which is known as DNA helicase, this helicase carry out, it can cause unwinding of DNA. It causes unwinding of DNA and it is ATP dependent. And it is ATP dependent. Now when if we cause it is a helicase enzyme which carry out unwinding of DNA, then the unwinding of the DNA molecule into two strands result in formation of Y-type structure like this. Y-type structure in these are the strands, and here these two strands which were joined complementary to each other now separated due to the helicase enzyme. So helicase enzyme inserted here, and when it runs, it will break the hydrogen bond between the two strands. So suppose this is the helicase enzyme. This causes unwinding of DNA and this Y-shaped structure is formed. This Y-shaped structure is known as replication fork. Is known as replication fork. So this way the replication fork is produced. So the replication does not initiate randomly at any place in the DNA. There is a definite region in E. coli where the replication starts, and we call it as the origin of replication. And at a such a region termed as origin of replication, we have discussed. Now in origin of replication, what will happen? After this, origin of replication. Here, as we discussed, suppose the origin is happening here. Okay, then it will cause a unwinding of DNA like this. So if a long DNA molecule is there and the two strands of DNA cannot be separated throughout the entire length of one stretch, the replication occurs within a small opening of a DNA helix and referred as a replication fork. So here you can see on this side if replication is going on. If this is a circular DNA, then the bidirectional replication will occur. Replication will occur on this side, on this side also. So these both Y-shaped structure are known as replication fork. So first what is going to happen? Replication fork is going to be produced. And after replication fork, suppose this thing is there, and this is 3 dash 2, suppose I am writing this is 5 dash to 3 dash, and this is 3 dash to 5 dash. Then these two strands, these are the single strands. These two single strands are stabilized by SSB protein. SSB is single stranded binding protein. Protein, and this single stranded binding proteins stabilize single strands of DNA. Stabilize the single strands, and that proteins are known as unwinding. Now when this unwinding take place and replication fork is produced here, at this back side you will see that the supercoiling develops at 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 end of DNA end of DNA opposite to replication fork at an end of DNA opposite to replication fork, supercoiling develop, and this supercoiling is raised by this supercoiling is released by DNA gyrase in prokaryotes and DNA topoisomerase DNA topoisomerase in eukaryotes. So this is released by DNA gyrase and DNA topoisomerase. DNA gyrase in prokaryotes and DNA topoisomerase in the eukaryote. Now, after this, single strands are stabilized and the supercoiling at the opposite end of the replication fork is released. The particularly here primase enzyme will come, and this primase enzyme will add a primer. So primer is just a short stretch of ribonucleotide which is synthesized in this direction. Okay, and that is opposite to this. It means it is in the 5' to 3'. Same way, see, suppose this much DNA replication fork is opened up to this side. So this RNA polymerase is getting RNA primer getting added, because primase will arc in always in 5' to 3' direction. So here the 5' to 3' direction is like this. So suppose the DNA strands are separated by helicase up to this point. This particularly RNA primer will added like this. When the DNA strands separate up to this point, like this, the RNA primer is going to be added in 5' to 3' direction. Okay, so this are the RNA primer which are added in this way up to the 5' to 3' direction. So like this RNA primer are added. Now this RNA primer, if we talk, dear student, this RNA primer are added by primase enzyme. And this RNA primase enzyme add this short stretch of RNA that is known as, or short stretch of ribonucleotide that is known as RNA primer. Here also this is RNA primer are added. Now RNA, after addition of RNA primer, why there is a need of RNA primer addition? Because there is a particularly to synthesize another strand, DNA polymerase is required, and DNA polymerase is somewhat large sized particularly protein which need a double strand to get stabilized. So there is a need of this RNA primer addition. So new stretch of DNA synthesized by a DNA polymerase 3, and this DNA polymerase 3 incapable of initiating DNA synthesis, or it is unable to deposit the first nucleotide in a daughter strand. So therefore another enzyme primase is required which synthesizes short primer of RNA, and this primate strand serve as a stepping stone to start errorless replication or to initiate a replication. Once the initiation of DNA synthesis is started, then RNA primer RNA strand is then removed enzymatically. So it will be removed later on. So how after the addition of this RNA primer, now what is going to happen? Let's discuss. Now here DNA polymerase 3 will synthesize the strand. Will synthesize strand. So in this, okay, and RNA primer is like this. Now here what will happen? The DNA polymerase 3 will sit like this, and this DNA polymerase 3 will add a ribonucleotide like this, and it will continuously synthesize DNA strand. But there particularly if it goes on continue particularly growing like this, and the particularly this DNA strand get elongated. So DNA dependent DNA polymerase 3 catalyze the polymerization only in one direction, that is the 5' to 3' direction. So it add the nucleotide in a 5' to 3' direction. Okay. Now if we continue discussing this further, we will see that this form a short strands of DNA, short strands of particular DNA that will be known as Okazaki fragments. So if we go on further telling, now here there is a RNA primer and this is a DNA strand. So this RNA primer and newly synthesized DNA strand are getting attached now. But there, if RNA ribonucleotide is present in the DNA strand, then it is considered as one type of mutation. So there is a need to remove the RNA strand. So now further, if we talk, then there is a need to remove the what RNA primer. So now further, if we talk, then RNA primers are removed by DNA polymerase 1. Now, when the DNA polymerase 1 removes the RNA primer, the DNA is going to look like this. So, these are the gap. These are created due to removal of RNA primer. Now, this gap are again filled by DNA polymerase 3. Gap are filled by DNA polymerase 3. And when DNA polymerase 3 will fill gap, it will sit here at this position and it will synthesize the further strands of DNA. So when DNA polymerase will fill the gap, the DNA will look like this. After the filling gap, you will see that like this strands are getting doubled. And here you will see the one bond is missing, and this one bond is missing, that is known as nick. This is known as nick. So nick is the nothing but it is a just a absence of single phosphodiester bond between the two Okazaki fragment. These two fragments are known as Okazaki fragments, and it is known that this nick is filled by DNA ligase, and DNA ligase will fill this nick. And when this nick will be filled, This new strand is in 5 dash to 3 dash direction. Here also this new strand is in 5 dash to 3 dash direction. Now this strand which is going continuously synthesized, it is known as this synthesis strand in the continuous one. This is known as leading strand synthesis. And on the another strand, the DNA synthesized in fragment on a discontinuous way, that is known as the lagging strand synthesis. And when ultimately DNA replication will be completed, the two copies of this DNA will occur like this. One is 5 dash to 3 dash in direction, and this is in 5 dash to 3 dash direction. This is a newly synthesized strand, whereas this is in 5 dash to 3 dash direction. So this is 3 dash to 5 dash direction. So this synthesis will occur in like this, that is a 5 dash to 3 dash direction. And these are the two daughter DNA molecules. In this way, the DNA replication occurs. Now in eukaryote, this DNA replication we have studied where the DNA polymerases are involved, ligase is involved, and all this occurred in DNA polymerase. But in DNA in eukaryotes, the replication of DNA takes place, and there different five types of different enzymes are required. Eukaryote replication, there is no need to study it in detail because it is not in our syllabus, but you need to remember some of the enzymes. So in eukaryote, five types of DNA polymerases are used, and that is DNA polymerase alpha, beta, gamma, delta, and epsilon. These five types of DNA polymerase are used. And in prokaryote, we have studied that the synthesis occur in two ways. One is in leading strand or continuous strand synthesis. It is fast because it is happening with the help of just single RNA primer and single DNA polymerases. But in the synthesis of leading strand or discontinuous strand is slow and requires many primers. And that is the DNA replication in case of the prokaryotes and the enzymes which are used in eukaryotes. Okay, I hope now you understood this, that how DNA replication occur in case of the prokaryote. So it is a mechanism of DNA replication. Now the experimental proof of semi-conservative mode of replication, it is given by Messrs and Stahl, that we will discuss in the next lecture. Thank you.